Hey, it's great to be back with another episode of Behind the Science with Neal here today, and we're going to be talking about his and his team's recent work on the Phase 2A open label trial of Dani in patients with DCM. So, Neal, I'll let you summarize a little bit of at a high level, your thoughts on the paper and the main results.
Yeah, thanks, Adith. I think it was pretty exciting. We tested the hypothesis that a myosin activator, Danicamtiv, would improve cardiac function in patients with sarcomeric dilated cardiomyopathy. So those were patients initially with MYH7 or myosin encoding gene variants that cause dilated cardiomyopathy. And the idea is if we improve myosin function and sarcomeric function, that these patients might be particularly prone to benefit from that pathway. We expanded the study in stages to include people with Titin gene variants, which also affect sarcomeric function, and then ultimately a third cohort, patients with variants in other genes or no clear identifiable genetic cause, which gave us a sense also for perhaps a sense of whether Danicamtiv would be more beneficial or have greater effects on contractile function in people with sarcomeric disease. And so in a small phase 2A open label study, patients received Danicamtiv for 2 treatment periods. Roughly, it was over a 10-day period of time. They got an echo at baseline, they got started on drug, they got an echo halfway, and based on the change in their systolic ejection time, which we used as a metric of Danicamtiv function, the Danicamtiv dose was adjusted. They got an echo at the end of the study. And what we present in the paper are the findings over that period of time. So not just changes in echo, but safety, tolerability, and some biomarker assessments of change with Danicamtiv therapy.
So I think, one thing to point out is these are clinically stable ambulatory patients, primarily NYHA 1 and 2, right? And so in terms of severity, not on that kind of end of the spectrum. And then you pointed out the sort of genetic component. So, you sort of, we're looking at this MYH7 cohort and the TTN cohort, and then the other cohort as well, which is this, the patients who don't fit in any one of those two categories. So I guess I'd love for you to comment on, we saw a benefit in LVEF for all groups, right, across the study, but definitely lowest in the other group, followed by the TTN, and then highest was the MYH7. And so, why do you think that was sort of speculative comments here, would be awesome?
Yeah, so again, in a non-randomized open label study that's small, we just have to be measured in how we interpret the findings. But what was intuitively appealing for the study from the beginning was to test the hypothesis that a drug that treated the underlying cause of disease might actually lead to the greatest benefit in people where their sarcomeric function starts out as abnormal. And we know this from observational studies in people that even before dilated cardiomyopathy develops, people who carry variants in these genes have modestly depressed contractile function using things like strain or tissue Doppler imaging. We know this in animal models and even in vitro studies that these MYH7 variants that cause DCM depress contractile function. And so the idea that we got an outsized response in the patients who maybe have their most proximate problem of sarcomeric dysfunction or decreased sarcomeric function aligns with that hypothesis, but needs to be reproduced, and I think we need to see it play out over a longer period of time.
And then I guess, talking about the changes in each group, right? What is that threshold for you as a clinician? You see a patient with DCM, maybe they have an MYH7 mutant or something like that. What is that threshold at which it becomes like, okay, this is something that is worth it, right? Because I think 9% was the upper bound in terms of absolute change, and even the other group had about a 4% change, right? So I think, it'd be great to hear how you think about that, putting your clinician hat on.
Yeah, it's such an important question. I think it's something that comes up every time we think about designing studies for people with very rare diseases, because you can open up the New England Journal and look at the latest heart failure trial in thousands or maybe even tens of thousands of patients. And practically speaking, that's just not possible with the rare disease space. And so a big question is, what's the surrogate endpoint that truly aligns with clinical improvements that patients and providers care about? And an ejection fraction improvement of 9% feels like a lot to me, but I think we want to see how that aligns with other metrics of efficacy. So do those patients have improved functional capacity? Do they feel better? Do other elements of cardiac function by echo look better? I think those are the things that we want to see, but that number to me feels meaningful and gives some hope for what next steps might tell us.
And then you were saying about, correlating that with the other, functional status things. And you guys did mention KCCQ 23 in the paper, and you guys kind of looked at that patient-reported outcome. And there's this movement sort of, I think, broadly to sort of start, using patient-reported outcomes more and more. John Spurtis is one of the guys who has been big in the field here. So I think, there was a trend towards it, but I think there was missing data and that not all the patients received the KCCQ. So is that something that you look for, and that you would use to guide you? How are patients responding? Maybe it's 3%, but they're saying they're feeling a lot better, and that's really the standard here.
Yeah, I think it's a really important question. So you pointed out, I think rightfully at the beginning, that this was not that sick of a patient population. They were mostly NYHA functional class 1 and 2. Their ejection fractions were low, and it allowed us to test the hypothesis that this drug could make contractile function better. But their NYHA functional class wasn't bad. Their NT-proBNP was also fairly low. And so had we even obtained KCCQ in everyone, I'm not sure that the results would have been different. This was a study that, like a generation of studies, took place over COVID. And so there were a number of protocol amendments, and part of the reason we didn't get a KCCQ in everyone in this study is that was a protocol amendment that happened after some people had finished the study. And I think for next steps, it's going to be important to understand how a patient-reported outcome aligns with these other metrics of efficacy. And KCCQ is the coin of the realm from the patient perspective, right? I think NYHA adds some information, but I think that'll be an important component. And then as a community, we can decide to what extent that people feel better when making a decision as to whether or not we want them to take a fifth drug, for instance.
Right. Yeah, because these patients, and we should mention, were on GDMT and they were stable on their GDMT, and then they were given Dani on top of that. And so I think one thing that I was interested in kind of reading the paper was their tropes increased for 3 patients, and those 3 patients all had, sort of non-MYH7, non-TTN variants. And you guys did see that trope increase, and you commented on the fact that it sort of went away, it was transient and went away on its own. It didn't really, need any sort of special intervention. And, I'm, as an outside person looking in, that's interesting to me. It sort of maybe even speaks to the fact that Dani is, sort of, selective for people with the variants that we think are, specifically going to be, most beneficial in this patient population. It's an interesting observation.
Yeah, and I'm glad you honed in on that. I think in the echo lab, these diseases seem the same, right? You got an echo, LVEF is down, LV and diastolic diameter is up, but we know at multiple levels that they are different diseases, that one cause of dilated cardiomyopathy may play out very different than another, and that a drug or just their baseline characterization would identify high troponin levels in one group of patients but not in another makes sense to me. And it's something that we need to watch carefully in the next stages of the development of Danicamtiv, is to understand not just differential efficacy, but the potential for differential toxicity. And while Danicamtiv and omecamtiv are different drugs, they're very similar. They're both myosin activators. And we know from a much larger experience in GALACTIC, for instance, that some degree of troponin elevation occurs with this class of drugs and hasn't necessarily associated with clinical events. And so it may not speak to myonecrosis, and I don't want to be ignorant to the possibility that could be a signal of harm, but we should also see how it tracks with other harm markers.
Yeah, for sure. That makes sense. And I think it was just an intriguing observation because it only happened in the other group, right? And I think, forward looking, I guess there are 2 comments here. One is like, how many patients with DCM do you think are sequenced today in the, at maybe tertiary medical centers and then also in the community at large? And that's my first question. So we'll start there.
Yeah, so I would say not enough. Genetic testing is pretty underutilized for dilated cardiomyopathy. We don't have great community data. There's some data using claims. So looking at a big insurance claims database and aligning an ICD-9 or 10 code of DCM with the use of genetic testing. And it would argue that the use of genetic testing for DCM is less than 5%. If you look at our center or at another referral center where we've had a genetics clinic for a long time, the number is going to be much higher than that. And I think there are things as a community, as healthcare providers, as really systems of care that we should do to make it easier for a cardiologist who wants to do this for their patient to get it done for patients. So it's just not used enough and should be used more, because I think you'll make decisions in 2025, not just about whether or not they go into a clinical trial, but you'll make decisions today that will help take better care of your patients with that genetic information. You'll be better at risk stratification. You'll be better at predicting progression of disease.
Yeah, absolutely. I think, enabling that is going to be like the future, right? And I think, one thing that's kind of top of mind now is gene therapies, and gene therapies for, DCM could possibly be conceptualized, right? Because we, in a number of cases, know what the pathologic variant is, and so sort of, if you have any thoughts on plans or things like that in the future that you want to comment on here with respect to gene therapy.
It's coming. Gene therapy for cardiomyopathy, as you know, has already been trialed for some very rare conditions. The first group of diseases being targeted are cardiomyopathies caused by haploinsufficiency. You're kind of missing half the amount of protein because you have a gene that's essentially knocked out, or at least you're haploinsufficient. You're missing half of it because one of the copies is truncated, for instance. So for people with that mechanism of disease, we can use something called gene replacement therapy, where you use a viral vector, you put the gene construct in there, it hones to the heart and delivers what is insufficient. For MYH7, that's not going to work. It's mostly a missense variant disease. And so just giving more MYH7 probably won't help. For Titin, it currently can't work because Titin is just way too big of a gene to fit in the viral vectors that we're using. And so I know people are working on things like gene editing, which might allow us to, say, fix a missense variant in MYH7, and different approaches for Titin, but I think those are a little bit further away. And where we're going to see gene therapies hit first in cardiomyopathy will be for those haploinsufficient cases. So I'm thinking of things like BAG3, PKP2, where there are already clinical trials running, which is ARVC, MYBPC3. Yeah, it's a hypertrophic cardiomyopathy disease gene. Clinical trials are going. And we'll learn a lot as we go that will not just be useful for people with those diseases, but I think could be generalized to other patients with cardiomyopathy. So it's here, closed clinical trials. Some of them are actively enrolling. So we're at a pretty exciting moment for our field.
Yeah, and then finally, if you wanted to share plans for Dani in the future and where you guys see this program going.
So we launched Kinship DCM, which is a phase 2b study of Danicamtiv in a very similar patient population. The details of Kinship DCM are on clinicaltrials.gov. But I think it will allow us to test the hypothesis that Danicamtiv improves cardiac function, but also improves how people feel, and I think would be part of a broader development plan. That's a study I'm helping to lead with Dr. Jim Januzzi, who's at BAME, and with the sponsor, Cardigan, who've been involved in the final stages of the phase 2A that we discussed today. That'll be an international multi-center randomized control study.
That's really exciting. We'll all be tuned in, waiting for those results in a bit. Thanks for your time today, Neal. I really appreciate it.
Thanks for having me.